So I know many of you know our candidate today, but I want to introduce Dr. Rusch to those who may not know Dr. Rusch. So I'm going to give a very brief introduction of highlighting some of her teaching experience, and I know Hannah is going to also provide some background information as she begins her seminar. So I don't want to double dip into her presentation of herself, but as I said, Hannah finished her PhD here at the University of Florida in 2023, and she worked with Dr. Wilson, I'm sure it's going to be ready soon, on the diversification of subtropical pastures to promote production and other ecosystem services. But in addition to being a graduate student in a laboratory, many of you know she was very, very engaged in lots of activities, including being a TA for many of the courses, and not just sort of grading, but in many of the experiences that she had at UF as part of being a TA, she actually had the opportunity to take a lesson or two, sometimes multiple lessons in courses where she was really in front of students and delivering content and activities in that capacity. In addition, I was really impressed, and so was the committee, in terms of some of the certificates she earned as a graduate student at the University of Florida that focused on teaching, because she was very passionate about that. And that included just some of the organizations outside of maybe the College of Agriculture that are at the UF level, where they really encourage graduate students to get more experience learning about teaching in case you're interested in a faculty position that might be like 100% teaching like this one, or teaching as a component of a tenure track position. In addition, she's done a lot of invited class talks all throughout, from undergraduate through her graduate experiences, both here at the University of Florida and the University of Minnesota. So the committee recognized her passion for teaching, and that's why she's here today to give this presentation. I really appreciate you coming back and seeing your friendly face and engaging with us today. So thank you, Hannah.
Thank you, Robert. Students, thank you all who are here in the room. Those of you online too, thank you for tuning in. I'll go get right into it. So I appreciate the kind introduction, and thanks to you all. All right, so I have been asked to provide a brief introduction about myself, but the bulk of the time today will be spent talking about photosynthesis. So we'll be doing a class demo. There will be some engagement that I'll ask for your participation and some learning activities today. But then there will also be time reserved for questions at the end. So my background is quite broad in plants and related sciences, going back as far as 2004, which is incredibly 21 years ago. Ecology and natural systems really caught my attention in high school, and this led me to pursue environmental sciences as an undergraduate. And through that program, I became very involved in natural resources management through agriculture, and the impact that agriculture has both on the environment, but also in terms of food security. So after I graduated, I became an AmeriCorps Vista volunteer. I was working as a liaison between the College of Food and Agriculture and Natural Resources Sciences at the University of Minnesota, along with community partners, organizations working with youth in urban agriculture, and help to create win-win solutions where university students could get internships in the community and communities could have access to university resources. I grew up speaking Spanish and added on Portuguese in undergrad, so I went south to Costa Rica, where I spent 5 years directing the President's Office and Board of Directors. While I was there, I was living on campus and embedded in their educational model, which is student-centered and experiential. So the campus is located in the humid tropics of Costa Rica. This is where I was living while I was there and working. My role was highly administrative, working directly with the presidents and board, and as chief of staff for the university, so working with the faculty of 40. But I also, in my free time, classes were Monday through Saturday. So Saturday mornings, I went to the field and you can see me here on my very first day of work at Earth, actually sorting solid waste. So that was one of the modules that was on campus: compost, organic farming, tropical cropping systems, tree nursery, ornamental nursery, animal husbandry. I cycled through all of them, and everyone was very surprised when I showed up at first until I just kept coming back and then it was normal. But I have been told no other assistant to the president has ever done that activity. I also was involved in providing extracurricular activities. All students live on campus. It's about 400 students from around 40 countries, mostly in Latin America, the Caribbean, and Africa, including a graduate here in the classroom. Sorry to call you out. But I was involved providing, for 5 years, I led English conversation groups every Saturday evening for 2 hours. Folks wanted to practice their English conversation skills. I also, on demand, delivered singing instruction and yoga instruction. So that was a very transformational experience and actually led me to graduate school. I knew when I graduated from undergrad, I wanted to pursue something, but I started as a vocal performance major, was environmental science, Spanish, Portuguese. I was kind of all over the place. But I brought everything together with agroecology, my interest in ecology, my interest in agriculture, and have spent the last 8 years in agricultural research here at the University of Florida for my PhD and for my master's, and now currently as a researcher in the Agronomy and Plant Genetics department at the University of Minnesota. So just to share a little bit about what I've been researching during this time, I'm currently working primarily on alfalfa, doing some work to develop protocols to do in vitro digestions of stem cross sections to be able to look at lignin development across time and across genetic lines of alfalfa. So shown here is 0 hours of incubation and rumen fluid. I do go and collect every Monday rumen fluid from a cow, up to 96 hours. So Dr. Sollenberger, I'm in your equivalent up there. Here in Florida, I was working in warm season perennial pastures, integrating legumes and winter annual grasses and legumes into our pastures here in Central Florida, and then in Minnesota, I got my start in agricultural research by integrating folks online.
I do have a coupon for it, but I'll take your insurance tax that out. So it'll be $7 for each medication. If you'd like to have me send that to her.
Yes. Will you just bill me for those or?
Yeah, so I would just need would you be shipping to UPS?
Okay.
And then for the copay here, I just need to collect the
We cannot hear.
Sorry, folks, how long have you not been able to hear?
Just since everyone was muted, but please go on. Sorry for the chaos.
All right, so everything from getting some hands-on experience in the field to producing posters and writing proposals to become a university scholar through the Center of Undergraduate Research, for example, and writing letters of recommendation afterwards for a couple of students, including current students in the program here at UF. I've also been involved outside of the university, delivering workshops as a community garden member, composting, leading interpretive naturalist walks, and as I mentioned before, I teach yoga. So my ambition and hope as an instructor is to inspire curiosity and empower learners using active learning, mentorship, and research. And so I try to bring these elements into everything that I do. And to give you a flavor of that, let's transition to our class demo. So imagine now that you are in the second half of spring semester, you came back from spring break. It's plant science, principles of plant science you're enrolled in. And we've already done the first unit that was very introductory, high-level overview of plant science as a field, moved into more plant specific structures and things to become familiar with what is a plant. And now we're in plant processes. So processes like photosynthesis before we move on to plant systems, which will be the final unit of the semester. So with that brief introduction and transition, welcome class. I hope you had a great afternoon. And we're going to get started with getting some juices flowing about our topic today of photosynthesis. So I'm going to invite folks who are online and in the classroom, take just 5 seconds and think about the first word that comes to your mind when you think about photosynthesis. All right, and now for those who are online, if you would type into the chat your response, and those in the room on the count of 3, say out loud, what was that word? 1, 2, 3.
Energy.
Okay, I heard energy in the chat. I see a couple chlorophyll, life. Okay, excellent. So you've got a good start, and I do see some people brought pen and paper into the classroom. That's great. Those of you online, go ahead and type in, and then at the end of the 30 seconds you have now, write down every word you can think about related to photosynthesis. So the next 30 seconds, please. Enter at the end of those 30 seconds. And all right, that's about 30 seconds worth of your time, folks. So turn to your neighbors now, next step, and share what words you had in common. Folks who are online, if you just type what your responses were into the chat, and then the leader to compile what responses appeared in multiple chats within the online. Take about a minute to do this. Yes. Wrap it up. Thank you. I hope that you've started your list here. Is anyone in the classroom want to share words that you had in common? Feel free to call them out. No volunteers? All right, I'll call the chloroplast. Chloroplast came up. That came up great. Wonderful. Thank you for offering. Folks online, thanks for typing your responses in. I'll read off some of the answers for folks here in the room. In common, that there was sugar, carbon, also carbon dioxide, light, and energy that they had in common. So this is great. Y'all have just gotten started on the first learning objective for today, which is to expand your vocabulary of photosynthesis as well as your comprehension of vocabulary related with photosynthesis. So we're going to go deeper into this today, but all of you already know something about photosynthesis. So this is a highly technical topic. We're going to stay at a pretty high level today. Don't be intimidated, but there will be new terms. So I hope that you get familiar with them and we'll dive a little bit deeper. So the objective is to, like I mentioned, stay at a pretty high level. So at least in general terms, by the end of today, I hope that you'll be able to describe the two processes, the light-dependent and independent reactions of photosynthesis and their separate phases. Additionally, we'll be practicing writing equations today using the chemical format, and we'll get some demos before I ask you to give your shot at it too. So be prepared for that as well. All right, so why do we talk about photosynthesis? And folks online, just making sure that slides are advancing and sound, you're able to follow along. Okay, chat,
All good, thank you.
Thank you. So plants form the first link in the food chain. This is important. Did anyone here have cereal for breakfast or drink milk, eat a product that was derived from an animal that eats a plant, or eat a plant ourselves? Probably all did, or I hope you did at least, and if you didn't, I'll get you a snack. So plants are capable of producing their own food, quote unquote, so converting solar energy into chemical energy that is stored in organic molecules and plant biomass. And this is what plants use to power their activities, but also what we as primary producers or grasshoppers, for example, cows, cattle, as primary consumers of these primary producers. So they are foundational to every organism within, or many organisms, in terms of where we get our energy, how we're fed. Additionally, plants produce oxygen. So photosynthesis specifically has a byproduct of oxygen, which shouldn't be too new, I think. But about 2.3 billion years ago, plants started photosynthesizing and increasing the amount of oxygen in the atmosphere, so that today we have oxygen levels in the atmosphere that are greater than CO2 that is respired by organisms like ourselves. So the humans hanging out in the corner of this diagram, for example, when we exhale CO2, but we inhale the oxygen that's in the air around us that plants are releasing through their leaves into the atmosphere. Another interesting thing is the source of that oxygen comes from water. Water taken up through roots in the soil, through plants is part of the photosynthetic process, as we'll see, and the oxygen is a byproduct of that. So plants are important, and photosynthesis specifically. As I mentioned, plants convert solar energy into chemical energy, sugars, carbohydrates, and this is a general equation that you may have seen previously. So on the left side of the equation here, there are inputs, so carbon dioxide, water, and the solar energy. This arrow represents a process, the whole process of photosynthesis, which we're really going to dive into today. And the outputs on the right side of the equation are carbohydrates and oxygen. So this is the kind of format that we'll be returning to throughout activity today, and you'll be asked to use this format later on. So wanted to make sure you had seen at least one example. So let's go into the two different processes of photosynthesis. There's the light dependent reactions, which as the name suggests, light is a requirement. And then there's the light independent reactions. That is the second part. And these two have interplay between one another as indicated by these arrows cycling between the two. So we're going to start with the light dependent reaction. But before we do that, let's just first make sure everyone feels oriented to where this is happening in the plant. So green tissues was one of the words that came out in the plant, or green plants rather, came out in the chat. And so we're zooming into this cross-section of a leaf here in the diagram. You can see there are different types of tissues that make up the plant leaf, and there are cells that are called mesophyll cells. Within the mesophyll cells are chloroplasts, and we had chloroplasts as one of the words that was shared in common in our opening activity today. So we're going to look closer at the chloroplasts now. Remember, we looked at this during the plant structure lecture in Unit 2, but we're going to pay special attention to certain features of the chloroplast today involved in the light dependent and independent reaction. So the thylakoids are these disc-like structures within the chloroplast, and illustrated here in this image, there is a yellow center in these thylakoids that have been cut in half. And that is what is known as the lumen. This is the interior of the thylakoid, and it's the site of the light dependent reactions. And then illustrated here is kind of just the space around the thylakoid. This this transparent liquid is the stroma, the area outside of the thylakoid where the light independent reaction takes place. Just before we move on, I have a question for the class. Our eyes perceive this chloroplast as green, and so I'm wondering if anyone would venture a guess at why our eyes might perceive these to be green. Anyone online or in person? You can feel free to chat with your neighbor too if you need a moment.
Sure.
All right, well, it's okay. You don't have to.
So
Thank
all
you.
that.
Hear that? I heard a very astute comment, so thank you for offering that. It is because the light is being reflected. That is correct. The visible wavelengths that are absorbed by chlorophyll, which are the pigments in the thylakoid membrane that absorb light for photosynthesis, they reflect the wavelengths that are green. Thus, our eyes pick up that reflected light. But it's between the 400 and 500 nanometer wavelengths and the 600 and 700 nanometer wavelengths that are absorbed by the plant. And as I mentioned, by the chlorophyll specifically. So these green dots here embedded in the thylakoid membrane represent the chlorophyll. It has this porphyrin head here, which contains nitrogen. And this is an important thing for anyone who has already looked into plant nutrition. You may know that the macronutrients that are important for plants are nitrogen, phosphorus, as we'll also see today, and potassium. This is one of the reasons why it's needed for the machinery of photosynthesis. All right, let's look a little bit closer at what's going on in the thylakoid membrane in our light-dependent reaction. So we're zooming in just again to orient folks. We're zooming into the thylakoid membrane. This diagram here, the arrow above is the stroma, that area around the thylakoid. The membrane that we were just looking at in the previous slide is represented here by this phospholipid bilayer across the middle of the screen. And then below the that membrane is the interior, so that yellow part of the lumen that we saw a few slides back. There are several terms that should be identified before we move on in this discussion. So things that I want to draw your attention to include photosystem 1, PS2, photosystem 2, and photosystem 1. It is a bit confusing. This one was discovered first, so it's photosystem 1, but it actually comes second in the process that we'll describe. The cytochrome complex is a pump that's going to be important for creating a concentration gradient, so be sure to be familiar with that. Just a little bit of review from chemistry. Remember, electrons are represented with the E superscript minus, hydrogen protons, H with the superscript plus, and this is inorganic phosphate, this P with the subscript I. Now, these might be unfamiliar for folks. Really, if you can remember NADPH, that's going to be good enough. Nicotine, adenine dinucleotide phosphate is a mouthful. So know that it's an electron carrier. And then ATP, this is an energy carrier, adenosine triphosphate. These are important things that we're going to take another step, diving a little bit deeper. So we'll walk through the diagram now. So the first thing that's going to happen is light is going to strike photosystem 2. This is going to energize an electron sitting at a reaction center, 680 nanometers. That energy from the sun is going to excite and displace this electron. It's going to go to a different energy level. In so doing, it leaves a void here that water is going to fill. Water is going to donate an electron to keep replacing that displaced electron. And in so doing, a process called hydrolysis, water splits into hydrogen and oxygen. This is the oxygen byproduct that we mentioned at the beginning of class. So the next thing that's going to happen, energy from this energized electron over here is going to transport down electron transport chain and drop off some energy, for example, at the cytochrome complex. And that is going to serve as a pump to take hydrogen from the stroma, the area outside of the thylakoid, and pump it into the thylakoid, into the lumen. So this is going to create a concentration gradient where there's higher concentration of hydrogen inside the lumen than there is outside in the stroma. So the electron, when it donates that energy to these different processes going on, it loses some of the initial solar energy, but it hits photosystem 1, and now more light is absorbed from the sun at 700 nanometers. It gets excited again, and ultimately it's going to catalyze this process of linking up NADP plus with hydrogen out in the stroma to form NADPH. And this is gonna feed into the light independent reaction, this electron carrier. Lastly, the concentration gradient of higher concentration here of hydrogen within the lumen is going to exit via the ATP synthase, an enzyme embedded in the thylakoid membrane, and the kinetic energy of diffusing across that concentration gradient is going to connect ADP and inorganic phosphorus from the stroma to create ATP, which is our energy carrying molecule. Any questions on this that folks would like me to spend a little bit more time on? Yes?
What's the FD there?
That's a great question. I'm going to gloss over it for today to keep things at kind of this high level. We can get into some more specifics in some of the videos on campus. All right, so now it's your turn. I'm going to have you connect with folks in your group and have you try to write out the inputs and the outputs of the light-dependent reactions. Again, staying at this high level. Feel free to use the diagram here. Folks online, you can consult with each other in the chat. And folks in the room, feel free to consult with your neighbors. I'll give you about 2 minutes to write out how you would express the light-dependent reaction using one of those chemical equations that we looked at the beginning of the talk today. Another 20 seconds or so, wrap things up. Come back together as a group. There, anyone either online or in the room like to share what you and your partner were able to come up with? Feel free to message in the chat. So, so the input are energy and water, and the output are oxygen, ATP, and NADPH. Thank you so much for offering that. And you got many of those parts right. So the pieces that are correct was the light energy and the water as well. And then on the outputs, we had the NADPH and the ATP. The only other thing that it wasn't explicit, but it is inferred by what the outputs are the NADP plus that combines with the hydrogen and the ADP and the inorganic phosphate that combine to form the ATP. So, great. All right, let's transition then to the like independent reaction, also known as the Calvin cycle or the dark reaction. You may be familiar with those terms. We'll talk in terms of like independent reaction today. So just to reiterate, this is receiving those inputs that came out on the output end of the light dependent reaction, the ATP, the energy carrying molecule, and the NADPH, the electron carrying molecule. These are feeding into the light independent reaction. And again, these are occurring in the stroma. So there are 3 phases of the light independent reaction, and we'll go through each one step by step. So the first one is the carboxylation phase or carbon fixation phase. And this is where atmospheric carbon comes into the equation. So we start with a 5-carbon molecule, and it's called ribulose bisphosphate. It has phosphate as well. This is going to be combined with CO2 with the help of the enzyme rubisco or ribulose biphosphate carboxylase. And so this enzyme is what catalyzes the fixation of carbon dioxide. It forms a 6-carbon molecule that's unstable, so it's immediately going to split into 3 carbon molecules called 3-PGA. And that is a brief summary of the carbon fixation phase, but something else important to mention, rubisco, which is very abundant on Earth because of its role in photosynthesis, is capable not only of combining with carbon dioxide, but also with oxygen. And this is considered a waste product because carbon, when it combines with carbon, it produces carbohydrates that then create biomass and the plant products that plants need and that tumors of plants use to get energy. But this process of photorespiration, which we'll see in our lecture next time when we talk about respiration, is considered a waste product. So Rubisco is another reason why nitrogen is important as a nutrient for plants, because as an enzyme protein, they're made of amino acids, which of course have nitrogen groups. So we'll move on to the next phase if there are no questions. Right, second phase is called reduction phase. If you remember from chemistry, reduction occurs when electrons are donated. So in this case, we have our inputs, the ATP, that energy carrying molecule, and the NADPH, the electron carrying molecule. They're going to donate electrons here in this reduction phase to convert, to create the energy to break and create new bonds between the 3-PGA to create this G3P molecule. And the G3P molecule is going to be the precursor to carbohydrates, other carbohydrate molecules. One of those G3P molecules is going to leave the cycle at this point in the reduction cycle. Like here you have some written notes you can take home with you of those processes. Any questions that I should spend some time on before we move on? All right, then the third phase of the cycle is the regeneration phase. And here we have the 5 remaining G3P molecules that needs to be converted back into the ribulose bisphosphate with the molecule, the 5-carbon molecule that we started with, the carbon fixation phase. So ATP is going to donate some energy to recreate those RUBP molecules in this phase. And there again are the notes for when you are looking back at this later. Anything you want me to cover, anything that was unclear here, you want me to spend a little more time? All right, great, because now it's your turn again. So folks who are online, I'm going to assign to you the carbon fixation cycle. On the left side of your equation, the inputs, on the right side, the outputs, just like we did with the lights dependent reactions. Folks over here on this side of the room, I'll assign to you reduction, and that leaves this group over here with the regeneration phase. So again, take another 2 minutes, consult with your peers, and at the end, we'll come back and share out just the individual phases that you're working on. Again, consult.
So, I don't know. Something like that. That's all I got to say. Oh, the way that.
I can things back together. Folks online, I'll ask for your participation first, since you had the first phase of the light independent reaction. Would somebody be willing to either come off, feel free to come off mute or use the chat to share? Okay, we have input was CO2 and HEP, and output was glucose. We also have a second answer that the inputs were the RUBP and carbon, and outputs were six carbon molecule. So you are on a good track. Yes, the RUBP and CO2 were what went in. And we did have the 6 carbon molecule, but then that quickly changed into the 3 carbon molecule, the 3 PGA. So representing it that way. Thank you very much, folks online, for your contributions. Folks in the room, let's go with the reduction folks first. Anyone here to offer up what you developed for the benefit of the class? And
There we go. Coming in 6 molecules of PGA and then ATP and ADPH. And then as outputs, we have 1 molecule of G3P plus the other 5 that goes to next phase, and ADP, PI, MDP, and the hydrogen.
okay, perfect, very good, nicely done. I appreciate you. That was the longest one, so they had a challenge. Thank you for working together too. And folks over here, can I have your rendering process?
Phosphorus and then hydrogen.
Done again. Thank you very much. All right, so you have achieved the task of summarizing all 3 phases. A global way of representing this might also be this equation, but you all got into even greater detail than the global. So this is really intended as a summary slide. Feel free to come back to it as you're preparing for studying for preparing for our exams. But just to highlight and summarize what we went over today, that global equation that we saw initially, light, carbon dioxide, and water go into photosynthesis, and out comes carbohydrate. Remember that the chloroplasts are the site of photosynthesis, where the lumen in the thylakoid is specifically the site of the light dependent reaction, and the stroma the site of the light independent reaction. And then you all have in your notes already the summary equations for each of those, since you graciously participated in the teaching activities. So be sure to refer back to those and back to here if you need to make any changes based on what you proposed and what's shown here. So with that, I'll give you a moment to write a reflection. You don't have to write all 4 of these prompts, just write to one of them. So you have a choice. Your first choice is to list and describe the new words that you learned today about photosynthesis. A second choice would be to think about, based on what you learned today, why nitrogen and phosphorus are needed in adequate amounts for optimal plant growth. Third choice, you can think about how drought conditions would impact photosynthesis based on what we covered here today in class. And lastly, feel free, if you choose, to describe the main differences that you see between the light dependent and independent reaction. Take a moment now to spend some time reflecting on any one of these 4 options. But again, just choose one. And you don't need to share this. And you can do more than one. Not limiting you. Yes?
Dr. Rusch, are you going to be collecting our reflections?
You can feel free to give them to me, but these are really for you to spend a moment digesting pretty technical comments, and then spend some time referring back to them if you'd like.
Can you offer extra points if you do more than one?
I think you're really the winner there, so. more than one, you'll probably get extra points on your exam and the quiz that we have after this, so.
It's good method acting, man.
Something right.
Here again.
People online can't hear us?
No, see the green.
Oh, it's good.
Now we can again.
Yeah, oh, the reflection just brought quiet to the classroom. Thank you for letting us know, though. I'm glad we're good. So, it's okay, yeah, we're approaching the end here. So, just to give you a heads up, what's coming up next, be sure to watch the video on C3, C4, and CAM pathways. You'll find that today what we discussed was primarily C3 photosynthesis, but especially here in Florida and warmer parts of the world, C4 is going to be a really important pathway for us to understand, and CAM photosynthesis is yet another way that plants have found to conduct photosynthesis. There's also a post-assessment quiz, so there's your chance at that extra credit, do well. And our next class will be on respiration. So feel free if any of you have questions, I'm here, I'm available to answer them. Folks online as well, you can feel free to unmute or type chat. With that, I conclude my demo of photosynthesis and open up questions for those in the audience, both online and in the room. Thank you. Cannabis. I should be able to hear you now. Want to ask a question?
Can I ask a question, Professor? For the lecture you just presented earlier, you mentioned the chlorophyll. There are two different types, chlorophyll A and the chlorophyll B. What are the differences between those two types of chlorophyll?
Yeah, thanks for your question. The chlorophyll A is involved with absorbing pigments at the 680 nanometers in photosystem 2. And the chlorophyll B is involved with absorbing wavelengths at the 700 nanometers in the photosystem.
Yeah.
Yes. Thank you for your question. Did that answer your question adequately?
Yes, thank you.
I know we have an exam next Friday, but I was gonna fly home and was wondering if I could take it early. Is that possible?
Nowadays,
I think it's a legitimate request, we'll consult with it, but you'll have to do the front of the whole.
still confused about the light dependent reactions. Why did we start with photosystem 2 instead of photosystem 1, etc.?
My understanding is that photosystem 2 was discovered second, thus they named it photosystem 2, even though in the process as we described it, it appears first. So that is counterintuitive, you're right, I agree that that is confusing.
Thank you.
And this can be like non-role play question asking. I'm happy to entertain questions from the class, I really am, and that was the logo of the presentation, so feel free to ask about the pedagogy, about anything that I skipped, maybe more electron carrier questions too. Yes.
So I found it very interesting, the beginning of the presentation, especially your experience at Earth. And how do you see that model of teaching, especially that here in, I'll probably generalize it's the American system, we've gone away from practical teaching, practical classes. How do you see that being incorporated? Do you see the value of that being incorporated? Would you do that?
Definitely, I think there are opportunities on this campus, and figuring out the logistics, I think, is going to be one of the obstacles. I don't know that it's an obstacle that cannot be overcome, but there are facilities on campus that I think would lend themselves to being able to get students engaged with plant sciences. Even as simple as going out to like Cardi Woods or going to look at some of these ornamental plants, especially in a plant like the principles of plant science class, not every student necessarily will become an agronomist. And so I think exploring many different types of plants would be an option. So I do think, and I would love to incorporate the experiential learning. Maybe they're not going to have a whole morning, 4-hour session developing skills in the pig barn or something like that, no. But we can find ways to take field trips, I think, to see some of the research that's going on, to use some of the instruments that are used to take plant measurements, like a chlorophyll meter, for example, is very accessible. And we could build data sets in class and try and do kind of this flipped learning too, where we go out and make observations and we come back with our data. We can look at what that data mean and how that data were derived. So I do see opportunities for it, and I would absolutely try to build that in to classes. I do recognize that Principles of Plant Science is offered online as well as in person. So using resources like the first resource through CALS to develop really high quality online learning, and getting creative about how folks can have meaningful, tangible experiences with plants and plant systems, even if they're not physically here on campus. So that would be like the next piece of the puzzle really to crack. But I do feel like there are opportunities to use resources on campus. Y'all have plants all year round here too that have active growth, which is really nice. So that would be another thing that I try to take advantage of in the courses that I work on. To build in as much engagement with students as possible. But I really do feel like in terms of building confidence and a sense of mastery, that it's important for students to feel like they have a practical experience. Even bringing plants into the classroom too, like and familiar with what grass they're walking over when they're walking on campus.
When you mentioned and your introduction. Some of that.
Yeah, well, one of the things that really got me interested in science was being in a classroom where a faculty member brought his research into the classroom. Very interesting ecological experiment, and he created an entire learning activity around it. And so I think experiences like that too, and things that I did as an PA were like bring a light meter into class when we were talking about yield formation, and share some of the results from my research where I had done some light interception research. So I think that there are ways to work with folks here on campus versus folks who are elsewhere who are doing interesting things related to the topics that we're covering, to try and pull in some of those technical components in terms of tools that are used, how they work, why they work, what their shortcomings are. Also, I would really be interested in finding a way to develop like a center for undergraduate course where you have students doing research in class. I think that would fit into potentially the skills area of the plant science, my major, excuse me, where they have one section of their program where they're required to develop field programs. I think that would be another avenue within the classroom, then also just helping students who are interested to explore options outside of the classroom too, to get involved in research. And scholarship on teaching is another thing, honestly, but I would be interested in that. I went to a presentation by one of the teaching faculty at University of Minnesota this year. That was really interesting, and one of my peers in the PhD program also did some teaching at scholarship. So that would be another realm that I think would be really interesting to. Another thing that this faculty member I worked with did like pre-assessment and post-assessment. It was so frustrating when you walked into the class, you're like, man, I don't know anything. Then you left the class, you're like, whoa, I learned a lot. And that's data that you can show to evaluate students in terms of what their growth was over the course of the semester. And that can be studied too. So I think things like those kinds of tools, and I know there have to be more out there that I'm curious to learn about, to see what our impact is as STEM educators, to really make sure that they leave the classroom not just with the degree that they came to seek, but really feeling they're confident in what they learned, what they
know. Over the course of your 21-year journey to this moment, you've kind of witnessed the transition from pretty much just in-person learning to the invention of online classrooms. COVID somehow forced us to try and do both at once, and hybrid environments can sometimes be difficult. Do you have any opinions or preferences for course development? Do you think that we should teach either online or in person? Is hybrid really tractable, or are we just trying to split our attention too much?
feel that know that I see your hand raised, so I'll address it after I address those questions. So yeah, I have seen that whole thing. I even worked as an undergraduate at a conference center, and there was this online university that would come for 1 week every year and like rent out the whole space. That was the only time they met. I was like, online learning, who would ever want to do that? But I have to say, I really appreciated the flexibility of online learning when I was a student here at University of Florida, especially having come from University of Minnesota, where there was none of that. And my closest research site was an hour and a half away. My furthest one was 3.5 hours away. And the other one I had to visit was 2.5 hours away every week during the summer, and starting here, like this part of April and into harvest season. So that was really challenging to try to manage the folks who are on site, their expectations, my faculty expectations that I would be in class, and then all the other things that I was trying to manage. So I developed a big appreciation coming to UF, and particularly because COVID did happened while I was a graduate student, like that infrastructure really allowed UF to pivot quickly and effectively. For myself as a graduate student, I think the effect for undergraduates or maybe even master's students may have been different. So I definitely see the need for folks who are working and doing certificate programs and want to pursue higher education, but maybe don't have the conditions to be able to step away into a more immersive setting. So I see the role and the need for the online learning. I do see the value, and I think particularly at the undergraduate level of the in-person learning, a little more structure, more skills based in a structured setting, I think, kind of setting experiments up for them to interact with versus a graduate student managing a little bit more on their own. And the hybrid format is a good question. Clearly, it has its challenges, you know, like I was muted and folks online couldn't hear me and I didn't take enough time to notice that before I advanced through a couple of slides. So that's a challenge and that's real. And I think being very attentive and making sure that folks feel included, especially if I can't see them with my eyes while I'm here in the classroom, unless, you know, maybe a voice comes and interrupts me. So I think that that would be the kinds of sensitivities that I would definitely need to have and develop and learn to manage really effectively to offer hybrid courses. But I've taken online synchronous and asynchronous courses and was involved in different formats of hybrid. Like when I was helping with the alternative cropping systems course, there was an in-person section that we recorded, and then this went online for folks who were in the asynchronous section. So I know that there's different formats and really finding what works for the different audiences. I do think there are different audiences and making sure that the materials that each one of those audiences consumes is contributing constructive towards their education. So I think using resources like first previously pulled CALs and other resources to develop effective pedagogy. And I would love to hear all of your ideas too. So what has worked and what hasn't worked, and I'll ask you during lunch. So
And there's one more question from Jean.
Yes.
Hannah, can you hear me? Yeah. If I remember correctly, you have served as a teaching assistant for the plant science course, right? When Dr. James Estrada taught that course. And my question for you, if you are going to like get this position and going to teach this course, is there some changes or modifications or some improvements you want to do for this course?
Thank you for the question. Yeah, I was the TA in the very first semester here at UF with James Estrada, and I went to every class section that first semester, and I learned all the 50 students' names, and it was really fun. I got to give a few lectures, including one on photosynthesis and respiration, which I was told was coincidental that that was the topic I was presenting on today, but it turns out it may not have been. But anyway, that's okay. To answer your question, because I was in the classroom each session, I think that there were things that I saw that I would keep, and there are things that I saw that I think we could change. So I really liked, for example, there was a lot of group work in the class. So every module, the students would form new groups and they would have a different kind of presentation so that they were practicing oral presentation, they were practicing scientific writing, they were practicing more like industry style writing. So I think things like that, where they're learning different skills and different formats, is helpful, and I would keep that element. I think that some of the things that I would change would be some of the lecture-based formats, incorporating more active learning opportunities within the classroom. because I think that one of the things that Canvas allows us to do is to have some, and granted, I know that there are demands on time spent. The time in the classroom is really precious, I think, in making sure that the students get a lot out of it and they get like connections to their peers and the faculty member in the room to try and really engage with the content. And then some of the things that's a little bit more didactic, things that could be communicated in like 15-minute videos and things like that, as kind of an assignment. I think would be something I would think to do to try and make in-person sessions, whether they be held online or in class, somewhat more dynamic, interactive with the material versus work at home being more reflective, studying of introductory reading, things like that. Does that answer your question?
Thank you. Can I ask you another question since I'm here?
Yeah, of course.
So do you think it is possible or feasible to add a lab component to this course?
Yeah, would it be possible to add a lab component to this course? It's a great question that I think we could find ways to add a lab to this course and decide what are the topics that lend themselves most to a lab kind of activity. For example, if we're looking at plant structure, maybe we bring plants in and we dissect them and we can look under a microscope to identify the different types of tissues and plant structures in different types of plants. Or if it's something that's a process, maybe like photosynthesis, again, using some kind of tool where we're measuring how effective a plant is. Maybe we could even bring in the C3 and C4 and look at like different levels within the different plants, carbon, amounts of carbon in the different tissues, like looking over results from a lab, for example. So I think that there are definitely ways that we could get lab components built into this course. Going out and visiting field sites, like I mentioned before, with some field trips to be able to look at like row spacing, for example, and why do we plant rows so far away from each other? What does that mean for light interception? What does it mean for weed management? Being able to go out and just see some of these concepts in practice, and finding spaces where that would be helpful in terms of learning, but also providing kind of more memorable that get them jazzed up.
Thank you, Hannah.